Hello, good morning everyone. Hello.
Hello, sir.
Good morning, sir.
Okay, that means you are hearing me, yes?
Yes, sir.
Yes, sir.
Now all of you raise your hands once. If this shows that most of you are active in your class now. Now I'm going to start the class. Okay, can you see my cursor also?
No, sir.
Yes, sir. Yes,
Yes.
sir.
Yes.
Yes. Okay. Now I'll start. Today is the new class, first class in hemoglobin and myoglobin. Okay. And in today's class, we'll give emphasis on the importance of these two molecules, hemoglobin and myoglobin, and we'll try to compare and contrast the structure and function of these two molecules. Just giving introduction to hemoglobin and myoglobin, we'll try to understand about their structure and try to correlate with the functions. However, more about the function we'll deal in our next class, but regarding the structure, we'll study in the today's class. Before studying hemoglobin and myoglobin, first of all, we have to know that why we are going to study hemoglobin and myoglobin. What is its importance? Why we are not studying about the other molecules, but we are studying about the hemoglobin and myoglobin, because as a medical student and after being the medical professionals, we have to know about the hemoglobin and myoglobin. Only then we can interpret a number of diseases. Now let's see some examples. This one, because the study of the structure and function of heme proteins provide information about these following points. Actually, hemoglobin and myoglobin, these are the heme proteins. In the classification of proteins, you might have studied like simple protein, conjugated proteins, and this is an example of the conjugated protein. And heme is the prosthetic group. That's why this hemoglobin and myoglobin are the examples of heme proteins, conjugated proteins. Okay, study on the structure and function of heme proteins provide information about the molecular basis of diseases like sickle cell anemia, thalassemia, which are related with the hemoglobin. We will study in detail about each of these disorders in our next class. But today we have to understand that before going into the sickle cell anemia and thalassemia, we have to know about the structure of these molecules. Only after knowing the structure of these molecules, we can better explain about the sickle cell anemia and thalassemia. Next is the mechanism of gaseous transport in our body. We all know that red blood cells are involved in the transport of oxygen and some about the carbon dioxide also. And these red blood cells consist of hemoglobin. Okay. And we also know that hemoglobin is transporting oxygen and carbon dioxide. But how hemoglobin is capable of transporting oxygen and carbon dioxide, this part we can easily explain after understanding about the structure of hemoglobin. Only after knowing the structure of hemoglobin, we can explain the structure and function relationship. Because of its structure, it is able to take the oxygen from the lungs and deliver to the tissues. Next is about the cyanide and carbon monoxide poisoning. What happens if carbon monoxide binds with hemoglobin instead of oxygen? Instead of oxygen, if carbon monoxide binds, what happens? This we'll study in cyanide and carbon monoxide poisoning. Next is high altitude sickness and adaptation to high altitude. When we directly go to a high altitude, then there is the problem in acclimatization. Yes, how, why there is the problem and how we cope up with that problem. To understand that, we have to understand about the structure of hemoglobin. This background information, hemoglobin and myoglobin are globular heme proteins, because you know the proteins can be fibrous, globular, and this hemoglobin and myoglobin are globular heme proteins. We already explained about the heme proteins, proteins containing heme as a prosthetic group. And the role of this heme group in this heme protein is dictated by the environment created by three-dimensional structure of the protein. You know that the structure of protein is usually complex. Most of the proteins are complex because there it has to create some sites, some environment for the chemical reaction here in hemoglobin and myoglobin to carry and release oxygen. Okay. We are talking about the heme proteins, and the role of heme in heme proteins is dictated by the environment created by three-dimensional structure of the protein. Okay. Heme protein, hemoglobin and myoglobin, we are repeatedly saying about the hemoglobin and myoglobin. In addition to that, other heme proteins, example of other heme proteins, just like cytochrome, catalase, these are also the heme proteins. For example, in cytochrome, function as an electron carrier that is alternatively oxidized and reduced. We are talking about the heme protein, and this cytochrome is also a heme protein. Cytochrome is involved as an electron carrier, or which passes the electron from one complex to the other complex in electron transport chain. That means there the heme group function as the electron carrier that is alternatively oxidized and reduced. And in catalase, this heme is a part of the active site of the enzyme that catalyzes the breakdown of hydrogen peroxide. We all know that catalase breaks down hydrogen peroxide, and this catalase is the heme protein, and the role of heme in catalase is to provide the environment for catalysis. Now our main hemoglobin and myoglobin, here heme group stops to reversibly bind oxygen. That means in the conditions of the lungs it will bind the oxygen, and in the conditions of the environment of the tissues it will release the oxygen. That's why we have said that role of the heme group is dictated by the environment created by three-dimensional structure of the protein. This was the background just to understand about the heme proteins. And while studying about the hemoglobin and myoglobin, we also have to know that there are other heme proteins too, which are important. It's like cytochrome and catalase are a few to know it. Okay. Why we are talking about the hemoglobin and myoglobin? Because, in brief, because they are involved in the supplying oxygen to our bodies, cells and tissues. Hemoglobin especially carry oxygen, myoglobin especially store oxygen within the muscle. They can carry within the muscles only, or store oxygen and release oxygen in times of demand, just like in vigorous exercise. Okay. This was the background information. Now we'll proceed forward with hemoglobin and myoglobin. First of all, functions slightly in detail than the earlier slide. Now hemoglobin. This is a globular protein present in high concentration in red blood cells. Globular protein, we have already said, hemoglobin present in red blood cells. We all know. Bind oxygen in the lungs and transport it to the cells. And in the cells, it releases oxygen, and again in the circulation, again it comes to the lungs, carry oxygen, goes to the cells, tissues and cells. That means and release oxygen, And this also transport carbon dioxide and hydrogen from the tissues to the lungs. We all know that oxygen, hemoglobin transport oxygen from the lungs to the tissues, and this all of us may not have given importance to this point. Also transport carbon dioxide and hydrogen from the tissues to the lungs, and how hemoglobin transport carbon dioxide and hydrogen will study in the later slides. And also carry and release nitric oxide in blood vessels of the tissues, especially in terms of the dilation of the vessels. This nitric oxide have a role, and this is carried by hemoglobin. Now the next is myoglobin. This is the oxygen storing protein present in the skeletal muscle cells. Release oxygen for use by muscle mitochondria for oxygen dependent synthesis of ATP under conditions of oxygen deprivation. That means hemoglobin carries oxygen, but myoglobin stores oxygen, especially in the skeletal muscle cells. Release of oxygen for use. They store oxygen and release oxygen for use in conditions of oxygen deprivation, because we have to study about a large number of metabolic processes. Some of them can occur even in the anaerobic condition, but most of the energy generated in the aerobic condition involving TCS cycle and electron transportation. For that, we need the oxygen. That means they are the aerobic conditions, and for those processes, this myoglobin releases oxygen in need. Now let's see the structure of heme. Sorry. Heme is a complex of protoporphyrin 9 and ferrous iron. We are talking about hemoglobin and myoglobin. These are the heme proteins. Now, first of all, we are studying only of the heme part. Heme is a complex of protoporphyrin. There are several types of porphyrin which are named according to the double bond present in them and the place of the double bonds. There are several porphyrin. We are not going to all porphyrin. However, in the synthesis of hemoglobin and in porphyrin, we will slightly study about the types of porphyrin there. A complex of porphyrin 9 and ferrous iron. That means we are talking about the heme, and the heme in hemoglobin and myoglobin, they contain porphyrin, that is protoporphyrin 9. And this porphyrin, here in the case 9, protoporphyrin 9, along with the ferrous iron, that is iron in +2 form, that in +2, Fe2 plus, and this is known as the heme. Protoporphyrin plus iron is known as the heme. The iron is held in the center of the heme molecule by bonds of 4 nitrogens of the porphyrin ring. There are 4 pyrrole rings, and those four pyrrole rings combinedly join to form the porphyrin ring, and each of the nitrogens join with the iron. The heme can form 2 additional bonds. Okay, we are talking about the heme now, and this heme is a part of hemoglobin, prosthetic part. Heme is the prosthetic part of hemoglobin, and in this heme it consists of protoporphyrin and along with iron in the center. Actually, this iron can bind with 6 different things. Let's say 6, like 6 valency. Now 4 different valencies are occupied by the 4 nitrogens, and now 2 are remaining. Among them 2, the heme can form 2 additional bonds, one on each side of the planar ring. We'll see along with the structure later on. In myoglobin and hemoglobin, one of these positions is coordinated to the side chain of histidine residue of the globin molecule, whereas the other position is available to bind to oxygen. Okay, this is the place where the oxygen binds in the heme. We're talking about hemoglobin and myoglobin. Hemoglobin and myoglobin are the heme proteins. They consist of heme as the prosthetic group, and in that heme contains porphyrin ring with iron in the center, and that iron binds with 4 nitrogens each of the pyrrole rings. And in addition to binding with 4 nitrogens, this iron can bind with 2 other things also, 2 other valencies, that means. And among them, one of these positions is coordinated to the side chain of histidine residue, This is one of the residues of the globin chain itself, whereas the other position is available to bind to oxygen. Okay, this is the place where oxygen is bind. Hemoprotein consists of heme, iron in the center, and iron can form six valencies. 4 different valencies are bound with each of the nitrogen of the pyrrole rings, which is forming the porphyrin ring, and now 2 valencies, one with the histidine residue of the globin chain and the other with the oxygen, and that after oxygen it will also bind with the histidine of the distal histidine. We'll see in detail later. Now this is the protoprofile. This is the heme group. This is the protoporphyrin 9 with iron in the center. This is the heme. We can see 1 pyrrole, 2 pyrrole, 3 pyrrole, 4 pyrrole rings forming porphyrin ring, and this is the type of protoporphyrin 9. And here this iron is in the center. This it can bind with 6 different constituents. 4 are the nitrogen of this porphyrin ring, and 2 with this is with the histidine of the globin chain, and on the next side it binds with the oxygen. This is the place oxygen is bound with the hemoglobin. Okay, this is the same structure, protoporphyrin 9. Okay. We are talking about the hemoglobin and myoglobin at a time because their structure is similar and their function is also similar. Not the same, but they're similar. That's why we are studying side by side. Hemoglobin is a tetrameric structure, you all know. It consists of 4 different chains. However, myoglobin is the monomeric unit. It consists of only 1 polypeptide chain. That's why, first of all, we'll study about the structure of myoglobin, which would be easier than the hemoglobin, because hemoglobin is a tetrameric structure, consists of 2 alpha and 2 beta chains, you all know. Okay, the structure of myoglobin, this is the conjugated protein. Apoprotein part, that means the protein part, the globin, a polypeptide chain of 153 amino acids. This number of amino acids is also important, especially when this MCQ examination started. Okay, hemoglobin is a monomeric protein, hemoprotein, consisting of 53 amino acids, and the prosthetic group, as you all know, this is the heme. Okay, this is the large myoglobin chain, which is one single chain, and in this heme group is shown here. And how it binds with oxygen is we have already studied. So this is the heme group, and this is the myoglobin chain. Okay. While studying about the structure of hemoglobin and myoglobin, we repeatedly study about the alpha helical structure, because most of the amino acids in hemoglobin and myoglobin, they exist in alpha helical structure. Around 80% of the amino acid residues are present in alpha helical structure. We all know, I've studied about the secondary structure of protein, that is alpha helix, beta plicate sheet, beta terms I've studied, and among them this hemoglobin and myoglobin, they almost all contains alpha helical structure. 80% of amino acid residues exist in alpha helical structure, which are present in 8 right-handed helices. And these 8 right-handed helices are named starting from the amino terminal as A, B, C, D, E, F, G, H. These alpha helical regions labeled A to H are terminated either by the presence of proline, because proline has the 5-membered ring which cannot be accommodated in the alpha helical structure. That means once the proline comes, that alpha helical structure is disrupted, or by beta bench and loop, which are stabilized by hydrogen. If there would be no proline or no beta bench, then all would have arranged in the continuous alpha helical structure, but continuous alpha helical structure is not possible because of this proline, and it is joined by the beta bond. That's why there are 8 regions which are named A, B, C, D, E, F, G, H starting from the amino terminal. The heme pocket is surrounded by E and H. That means we have seen the heme group, and this heme group is surrounded by E and F. Among the 8 alpha helix structure, E and F actually surrounds the heme pocket. Moreover, H is also near to it. We will see the structure. E and F, you can see this is okay. And now here, starting from the amino terminal, this is the structure of myoglobin, and this thickened the bold part, alpha helices are shown here. Okay, this is the alpha helix A, and here is the tone, and this is known as AP part. Now this is B, then some discontinuous structures, C, again some discontinuous D, and you can see the E, F, G, H, and among this heme group is just inner cleavage formed by E and F group. E and F is okay. H is also near, you can see. And side photo is taken from the different side, that's why this is slightly farther away. H is one, but this is actually nearer, just slightly farther than F, E and F. But this G group is more farther from this H group. Location of the polar and non-polar amino acid residues. The interior of the myoglobin molecule is composed almost entirely of non-polar amino acids. That means it will be easier if you think of the aqueous environment. That means if the aqueous environment, the outer aqueous environment, that means the polar groups containing amino acids will be on the outer side. That's why the interior of the myoglobin molecule is composed almost entirely of non-polar amino acids. They are packed close together, forming a structure stabilized by hydrophobic interactions between these clustered residues. In contrast, charged amino acids are located exclusively on the outer surface of the molecule, where they can form the hydrogen bonds and also react with the water. Okay, are you getting me? Are you hearing?
Yes, sir.
Okay, now binding of the heme group, we have already seen in the picture also. The heme group of myoglobin sits in a cleavage in a molecule which is aligned with non-polar amino acids. We have already shown. But all the amino acids can not be non-polar, in that E and F alpha helices also. Notable exceptions are the histidine residues. One proximal histidine binds directly to the iron in heme. The second, or the distal histidine, does not directly interact with the heme group but helps stabilize the oxygen binding of oxygen to the ferrous and iron. When stating the lines, there are no lines. Yes, okay, we have already said that the central iron can bind with the 6 different constituents. Among them, 4 is the nitrogen of the porphyrin ring, and 2 are, one is the proximal histidine, that means the nearer histidine. It directly binds with the proximal histidine, and other valence we have said it binds with the oxygen, and after binding with the oxygen, that is stabilized by this distal histidine. You will see with the picture also. Okay, one, this proximal histidine, it binds directly with the proximal histidine, but with the distal histidine it does not bind directly, but after binding with the oxygen, and this is just opposite side to that side, and it stabilizes the structure. The protein or globin part of myoglobin thus creates a special environment for the heme group that permits the reversible binding of one oxygen molecule. We are talking about the myoglobin. It has one chain, one polypeptide chain. What was the amino acid?
153.
Okay, one. Now this is the model of myoglobin showing the helices A to H. This you can see the helices A to H, and here once more about the bind. You can see this ring as the heme group, iron along with the protoporphyrin 9. This is the red part. Let's see the iron is shown in the yellow in the middle. Okay, among the 6 valencies, 4 valencies with this itself for nitrogen, and 2 valencies, one with the proximal histidine, it binds directly, one valency, and the other valency, it binds with the oxygen, and the binding with the oxygen is stabilized by this distal histidine. It is because of the charge of the histidine. This is stabilized by this histidine. Distal histidine means which is the histidine which is farther one. Proximal histidine means the histidine which is the nearer one. This is proximal histidine present in the F8. F8 means the 8th position of the F chain, and this is distal histidine. E8 means 7th amino acid of the E chain. Okay. This is about the structure of myoglobin. Okay, after going through the structure of myoglobin, now we'll go to the hemoglobin. Before studying hemoglobin, why we studied myoglobin is their structures are similar. Hemoglobin is a tetramer consisting of 4 different chains, 2 alpha chain and 2 beta chains. However, myoglobin is the monomeric structure consisting only one polypeptide chain. Now, the structure of the individual polypeptide chain of hemoglobin is not same but similar to that of the myoglobin, and the sequence of amino acids are also slightly different, not that much different. They differ slightly here in case of hemoglobin, and amino acid in alpha and beta chains are also slightly different, not that much different. And now these 4 chains are actually not coded by the single gene also, and after the formation of single individual chain, now this complex hemoglobin is formed. That means the structure is somewhat more complex than that of the mile. Conjugated protein, globin, apoprotein part, we all know heme is the prosthetic part, consists of 4 polypeptide chain and 4 heme groups. Okay, here you don't be confused. It consists of 4 polypeptide chains. It consists of 4 polypeptide chain means it also consists of 4 heme group. In individual polypeptide chain, there is also a heme group. As we have studied myoglobin first, each individual chain you can compare with the myoglobin. If you compare each individual chain with the myoglobin, then you will not forget that it consists of the 4 heme groups. That means the 4 chains contain one heme groups. Common form is hemoglobin A. I think there are different forms of hemoglobin. We'll see later on. We also know about the hemoglobin A, hemoglobin A2, fetal hemoglobin, and in this hemoglobin, all the hemoglobins are the tetrameric structure, but the chains are different. Some are alpha to beta, some are to alpha to delta. Like this, chains are different. And we are mainly talking about the hemoglobin A. That's why 2 alpha and 2 beta chains. Molecular weight is 64 kilodaltons. Okay, alpha chain consists of 141 amino acids and the beta chain consists of 146 amino acids. Okay. If you look the number of amino acids, we can say that the beta chain is nearer to the number of amino acids as compared to the MIG. Beta chain 146 amino acids. The alpha chain, both of the alpha chain consist of 141 amino acids each. However, the beta chain consists of 146 amino acids each. 4 polypeptide chains are held together by non-covalent interactions. Okay, you have studied about the proteins. You have studied about the structure of proteins. You also know the structure of protein is complex. That's why we study the structure in different hierarchies: primary structure, secondary structure, tertiary structure, and quaternary structure. Now you have studied in case of myoglobin, only it gives primary, secondary and tertiary structure, but not the quaternary structure. Because in quaternary structure you study how the different polypeptide chains are arranged with each other. In that thing we are studying here now. 4 polypeptide chains are held together by non-covalent interactions. That means this is about the quaternary structure. Each chain contains a heme group that binds one oxygen molecule. That means tetrameric structure, 4 globin chains, 4 heme group, and it can carry 4 oxygen molecules. Can bind with 4 oxygen molecules. But in the myoglobin, 1 oxygen molecule because only one chain, only one heme. Alpha chains and beta chains consist of 7 and 8 helical regions from A to H. What is this? Alpha and beta chains consist of 7 and 8. That means alpha chain consists of 7, whereas the beta chain consists of 8 helical regions. That means the structure of beta chain is identical to that of myoglobin. It also consists of 8 helical regions, but this alpha chain, you can see it is slightly shorter also, and this does not contain the D helix. There is no D helix in the alpha chain of the hemoglobin. Each subunit has stretches of alpha helical structure and a heme binding pocket similar to that described for myoglobin. That's why we have studied the myoglobin before going to hemoglobin. Each subunit has stretches of alpha helical structure. That means 8 different stretches A to H and a heme binding pocket similar to that of myoglobin, except that in the alpha chain there is no D helix. However, the tetrameric hemoglobin molecule is structurally and functionally more complex than myoglobin. Individual structure is same, but as a whole, the structure is more complex and function. To understand the function also, it is more complex. Furthermore, the oxygen binding properties of hemoglobin and myoglobin are regulated by interaction with allosteric effectors. Allo means different and the steric means the side, that means the soft chains or the substrates which bind to the different sites. That means the oxygen binding property is influenced by the interaction with the allosteric effectors. Okay, quaternary structure of hemoglobin. By this time you all know the hemoglobin is a tetramer and can be described as being composed of 2 identical dimers. Hemoglobin is a tetramer consisting of 4 chains, 2 alpha chain and 2 beta chain. Hemoglobin is also a dimer consisting of 2 alpha beta chains. Alpha beta 1, alpha beta 2. Why we are also saying as a dimer? Because this one alpha and 1 beta, they bind with somehow strong bonds, and another alpha and beta, they bind with somehow stronger bonds, but the connection between these two is slightly weak. That's why it can also be said as a dimer of 2 identical dimers. The 2 polypeptide chains within each dimer are held tightly together primarily by hydrophobic interactions. Now we'll see the picture also. In contrast, the 2 dimers are able to move with respect to each other, being held together primarily by polar bonds. That's why they are called as the dimer of the dimers, because one alpha and 1 beta, they are bound strongly. Another alpha and beta, within it they are bound strongly, but their connection between those is slightly weak. The weaker interaction between these mobile dimers result in 2 dimers occupying different relative positions in deoxyhemoglobin and in oxyhemoglobin. That's why when they occupy slightly different position, that means the structure of hemoglobin is also slightly changed in oxygenated and in deoxygenated state. That's why we talk about the taut state and the relaxed state. There are 2 states, taut form and the relaxed form. Okay, now let's see this structure. This is the hemoglobin tetrameric structure, as we all know. Now we have also said as the dimers, because this bond is slightly weaker and the bonding pattern is different in oxygenated state and deoxygenated state. Okay, let's see. This is alpha and 1 alpha and 1 beta. This is the next alpha and beta. and among between this alpha and beta and all this alpha and beta, there is the strong interactions, primarily hydrophobic interactions. That is the strong interactions. Strong interactions, primarily hydrophobic, between alpha and beta chains form stable alpha beta dimers. They are forming the stable dimers. Those two are forming the stable dimers. However, the connection between these dimers, this is the weak ionic and hydrogen bonds occur between this alpha beta dimer. That's why sometimes this tetrameric structure of hemoglobin is also said as the dimer of the dimers. This is the tau form or the deoxygenated form. That means you can see here also T or tau. This is the structure of deoxyhemoglobin. And when the oxygen binds with the hemoglobin, this binding pattern will slightly change. That means some of the bonds will be broken down and it will go to the relaxed state. That's why this oxygenated form is known as R or relaxed structure of oxyhemoglobin. Some ionic and hydrogen bonds between alpha beta dimers are broken in oxygenated state. After some of the bonds between this dimer of dimer are broken, this goes to the relaxed form. This is the tau form or the tensed form simply because there is a strong bond between them. However, after the oxygen binds, this is the loosely bound or the relaxed form. Okay, now let's see this plane. This we are showing about the protoporphyrin. Okay, this black lines, it shows you the porphyrin ring, and this is the histidine effect. That means out of the 6 valencies, 5th valency is bind with this histidine effect, and the 6th valence is bound with the oxygen. Now this is the deoxygenated form, and this is the oxygenated form. Now let's see this part, and when oxygen is added in this picture. Okay. This is the histidine of F8. This binds with the iron, that in the 5th position, let's say, because the 4th already occupied by different nitrogens. And when this oxygen from the other side comes and bind with this iron, let's say this is the 6th valency, and when it binds, this iron slightly enters into the plane of the porphyrin. That's why there is a slight change in the structure of oxyhemoglobin and deoxyhemoglobin. As you see, this iron, this is slightly farther away from this porphyrin plane. Now, when the oxygen binds, you can see it is in the same as the porphyrin plane. This iron comes inside this porphyrin plane. However, there is very little difference. This is the magnified portion. There is little difference, and due to this, we have also seen as the tau and the relaxed states. The binding of oxygen to the heme iron pulls the iron into the plane of the heme. Because the iron is also linked to the proximal histidine, there is the movement of the globin chains that alters the interface between the dimers. The binding of oxygen to the heme pulls the iron into the plane of the heme. Because the iron is also linked with the histidine, and now that histidine part, it will also be moved. That's why there is a change in the structure. Okay, we have already studied now this T form. The deoxy form of hemoglobin is called the T form or the tau form or the tens form. The two dimers interact through the network of ionic bonds and hydrogen bonds that constrain the movement of the peptide chain. The T form is the low oxygen affinity form of hemoglobin. Now the next form is R form or the relaxed form. The binding of oxygen to hemoglobin causes the rupture of some of the ionic bonds and hydrogen bonds between the dimers, and this goes to the relaxed state. Okay, oxygen is already bound. Why relaxed? That means after one oxygen is bound, another oxygen has to bound. There are 4 different oxygens to be bound with the same hemoglobin molecules, and it is easier for the second oxygen to bound with that hemoglobin when compared to the first, and it is more easier for third and even more easier for fourth. This is explained by the relaxed form. This leads to the structure called the R or the relaxed form, in which the polypeptide chain have more freedom of movement, and the R form is the high affinity form of hemoglobin. Now we have studied about the structure of hemoglobin and myoglobin and we have also listed about some of the points why we have to study the structure of hemoglobin and myoglobin. And the function we'll study in our next class. Binding of hemoglobin to myoglobin, binding of oxygen to hemoglobin and myoglobin is different. Myoglobin can bind only one molecule of oxygen because it contains only one heme group. Hemoglobin can bind to 4 oxygen molecules, one at each of the 4 heme groups. Okay, binding of oxygen to myoglobin and hemoglobin. Myoglobin can bind only one oxygen molecule because it has only one polypeptide chain, only one heme group, whereas the hemoglobin can bind 4 oxygen molecules because it is the tetrameric structure of 4 globin chains, 4 heme group, that means 4 iron. That's why it can bind with 4 different oxygen molecules. Now in our next class, we will study about the oxygen saturation curve. It is different, how the function of hemoglobin and myoglobin is different, why hemoglobin is a carrier molecule, that means it carries oxygen from the lungs to the tissues, and the myoglobin, why it is a storage molecule. We'll better understand by studying the function and the oxygen saturation curve in our next class. Okay. If you have some queries regarding today's class, you can just unmute your mic and put your questions. If mic doesn't work or you want to message, then it's also okay.
Sir, can you explain that proline part?
Okay, proline part, it is a 5-membered cyclic structure. That's why when proline comes in between the chains, then there is a disruption in the hydrogen bonds, which otherwise has to form in the alpha helical structure. If you study about the alpha helical structure in the protein secondary structure, you will come to know. I guess every fourth amino acid or some add, there should be the hydrogen bonding, and this will be disrupted once the proline comes. That's why the helical structure is not possible. That's why when the proline comes, that helical structure will be disrupted. If you go through the secondary structure, in the chapter secondary structure, you yourself also better know about the bonding pattern, but that is not possible when the proline comes in between. Attendance part. Okay, now I'll download your attendance. Hope most of you are already present. Okay, any more questions?
Sir,
Yes.
sir, can you please upload this slide?
Yes, yes. Sure.
Thank you, sir.
class also I have uploaded. If you have no further questions, this is today. Thank you, class.